Hello, my name's Robert.
I'm Joel.
I'm Maria. I'm Isabel.
I'm Gabe.
And welcome to our presentation about teeny tiny bacteria. So, what is a teeny tiny bacteria? Well, according to our definition, their genomes under 2 megabases. So just for comparison, the average bacteria genome is around 5 megabases, so around half the size. And the reason why we're interested in these groups is because oftentimes when you're doing environmental metagenomes, the smallest bacteria often are missed during something like a bulk sequencing step, because for the programs to assemble a genome, essentially, you need to hit a critical mass number of reads, and because they're often much smaller, we have less of their reads, and so we don't really hit that critical mass to get an assembled genome. And another thing is they're often endosymbionts, so there's just less of them compared to freestanding bacteria with larger genomes. And so we've developed a new method to really investigate these bacteria. So our soil samples come from Harvard Forest, which is a public forest in Petersham, Massachusetts. It's also a NEON site, so a lot of climatic and environmental research is done there, stuff like temperature monitoring, CO2 emissions, and also our soil samples. So from the soil samples, we introduced them into an increasingly fine series of filters from 20 to 5 to 1.2 to 0.2 microns thick. And then from there, we take the 1.2 and 0.2 micron filters and we extract all the biological samples from those. We introduce the SYBR green dye into them, and we use FACS to sort them into 100 cell wells. And from there, we do essentially mini metagenomics, which is a compromise between bulk and single cell sequencing. And from those 100 cell wells, we extract the DNA, we send them off to New England biolabs for sequencing. So once we get our reads, the first step we do is we trim them because the ends tend to be a little erroneous, and then the next step would be to assemble them into longer contigs, and then the next step would be binning. Essentially, we take contigs and we match them by similarity into bins, and you can think of each bin as essentially representing an entire organism. And there we can do a couple of things. The first thing we did was annotate them. So Proca looks for open reading frames in the contigs and matches them to corresponding proteins. And then after that, we can use a pathway annotator like KEGG. Essentially, the program will tell you which pathways are supported based on the protein expression in your genome. So you can say, oh, this bacteria supports glycolysis, but not the electron transport chain. Taking a more environmental and physiological view, we can sort all the contigs into one big set, and we run that through GTDBTK, which will compare our genomes against the entire database of GTDB, which contains all the submitted genomes across the world, and we can see, oh, our genomes are in this taxonomical branch of the tree of life. And I'll turn it over to Maria next.
So the group I have been studying is Microbiota phylum. This is a relatively not very classified phylum. The most well-known members are endosymbionts with flagellated protozoa in termite guts. This is actually the only environment where they are in high abundance. They're also found free living in the environment, but because they have smaller genomes, they're often left out of traditional metagenomic analyses, so there is not much known about these free living members. In our soil samples, we had 3 MAGs, which are metagenome assembled genomes. 2 of them had a completeness of over 60%, and these higher completeness members were studied. The first one had a 2.7 megabase genome, which is on the larger side of our data, and the second one had a 1.5 megabase genome. So far, the main finding has been that both of these MAGs have high carbohydrate active enzyme counts relative to the other similarly sized MAGs in our data, and we would expect this to be the case if they were endosymbionts, because in termite guts, they act to help the termites degrade lignin and cellulose. And this is a tree of the Illuci microbia class. Both of our mags are in.
Hi, so I chose the chlamydia samples that we found in our sample genomes. Out of all the bacteria that we found, 9 of them were chlamydia that had a completeness of greater than 50% and less than 10% contamination. Of those 9, 8 of them belong to the Rhabdochlamydiaceae family, which is the largest and most diverse family of the chlamydia class. The other genome is a member of the SM23-39 family, and all of these genomes are around 2 megabases, which is right at that cutoff for teeny tiny bacteria that we've classified as teeny tiny. And just for reference, the Chlamydia trachomitis genome is around 1 megabase. And so we're comparing these samples to the Chlamydia trachomitis genome mostly because we know that that's the most studied one out of all the chlamydia. And so we're comparing them to that to kind of get a better idea of how it differs from that specific human pathogen. And in general, we've found that these pathogens have a more complete TCA cycle than the Chlamydia trachomitis one, and they have other metabolic pathways for using other carbon sources, which indicates that it has less of a reliance on the host cell. It also has more amino acid biosynthesis pathways than the Chlamydia trachomitis, and it also has more antibiotic synthesis pathways, which is consistent with a soil lifestyle that we have seen in other soil microbes. It also has more secondary metabolite biosynthesis pathways, and just overall it has a larger genome, so it has more genes and more proteins that it encodes for than the Chlamydia trachomitis genome.
Hi, so I'm Joel. I'm going to be talking about the Pesticide-Bacteria superphylum. So I chose to study the Pesticide-Bacteria superphylum in our dataset because I thought it was really interesting. That was the most prominent group. And so as you can see right there, that is the phylogenetic tree of all of our Pesticide-Bacteria present in the dataset. So there are 47 total bacteria out of 177. And so there was the identification of 4 new classes, 11 families, and 11 genera. And so the average genome size was 1.34 megabases, while the largest was almost at 3 megabases, and the smallest was a little under 1. And so what is cool about this group is that there is a prominent lack of core proteins and genes, which then leads to them having a lower completeness in the genomes. And so we can make the assumptions that they're lesser metabolic capabilities, and so further studies need to be done, and that is what we're working on to see the existence of other core proteins and genes that might exist, and how that might be prominent on the existence of newer metabolic pathways that might not be identified yet in the different databases we utilize.
Gabe and I chose the Thermoplasmatata phylum in Archaea, and so in the Thermoplasmatata phylum we discovered 2 metagenome assembled genomes, and one of them was over 50% complete, and in that one that was over 50% complete, it's found to have 1.68 megabases, and it's 79% complete, so the projected size is around 2.1 megabases. And in the Thermoplasmatata phylum, there's not much known about the class that this organism has been found to be in or class E2, but the Thermoplasmatota and Posidonia have been found to be in various environments, including the ocean and the human GI tract and soil, as well as swamplands.